Thank you so much for that, Leon. My name is Charisse, and I'm a postdoc in Jacquin Niles' lab in BE. And together with Khan Osman and Maina Kashima, we're working towards transforming our ability to discover new antimalarial drugs with novel mechanisms of action. So every year, hundreds and millions of people suffer from malaria globally. Malaria is a curable disease, that's true. However, the emergence of drug resistant parasites has led to treatment failure, and this has really dampened our efforts to eradicate malaria. And this is especially problematic for the first line drug artemisinin in Southeast Asia. Of note, some regions in South America that has eradicated malaria in the past is currently experiencing resurgence of malaria cases. And then finally, the heaviest burden of the disease is still in Africa, where malaria heavily impacts both economic and healthcare infrastructures. Malaria is caused by parasites of the Plasmodium genus. It has two obligate hosts, the mosquito vector and mammalian hosts, as you can see here, such as humans. Infection of the human red blood cells causes the clinical manifestations of the disease, and this translates to symptoms including fever, chills, and in advanced cases, respiratory distress, cerebral dysfunction, and even death. And so, all of this really is to say that we need new antimalarial drugs with novel mechanisms of action. However, the antimalarial drug discovery process is a long and tiring process of hit to lead compound profiling prior to getting these compounds eventually into clinical trials, product development, and then eventually licensure. In addition, some of these compounds can advance through this pipeline without our knowledge of its mechanism of action at onset. And so this has really led to the licensure of combination therapies of currently used antimalarials with already diminished efficacy in some regions of the world. But what if we can evaluate or characterize novel compound libraries in parallel with target identification? This can allow us to deploy new compound target pairs with a potential for durable efficacy in the field. And so, following this approach, our proposed solution combines the strength of high throughput drug discovery process with in-parasite target identification and bioactivity profiling for antimalarial drug discovery. And so, how this works is that we can take any set of uncharacterized Plasmodium falciparum proteins with likely essential function. We validate this essentiality in the parasites using a TETR-based gene expression regulation tool that we've developed in the lab. This tool can be also used as a target-directed antimalarial drug screening platform. In parallel, we carry out heterologous protein expression in E. coli to be able to narrow down on proteins that we can express robustly and screen against various chemical libraries. And so taken together, these two routes allows us to carry out target-directed screening with large compound libraries, B, compress our timeline for antimalarial drug discovery, and then finally introduce novel compound target pairs into the market with a potential for durable efficacy in the field. So to execute this plan, we use our gene expression regulation tool, the TET-RDOSI RNA aptamer module, to then identify proteins in the parasites that's essential for parasite growth or expansion, which is summarized in this viability plot here to the left-hand side of the screen, with the essential proteins shown in blue compared to an unessential protein shown in red. And of these proteins, target number 10 localizes to the parasite's nucleus, as shown in this immunofluorescence assay. It expresses robustly in E. coli and is hypothesized to be a zinc binding protein based on the crystal structure as well as sequence similarity assessments. Now, screening of this recombinant protein against the 50,000 small molecule library from Angela Kohler's lab at the Koch Institute identified 9 compounds that bound specifically to our target protein. And so when the lab is up and running, we plan to carry out target engagement assays in the parasite using our functional genetics tool and differential sensitivity assessments. We also plan to carry out experiments with our recombinant protein to look for compound target binding affinity. And also, we plan to carry out structural activity guided synthesis to obtain lead compounds. And then we plan to repeat this whole workflow with the rest of the targets in our essentiality, with the rest of our essential targets, as well as screen these to additional compounds, either from the public collection or through our collaborators. And so finally, a visualization of our timeline shows that with our approach, we were able to characterize druggable targets in malaria parasites and identify inhibitors that are specific to one of these targets in a shorter period of time. Now we are geared towards carrying out target engagement assays in the parasite, as well as screen additional compound libraries, including the 2 billion compound library from Zebi AI, as well as public compound libraries from our collaborators and pharma. We also, in parallel, plan to carry out engagements talks with our market and customers to be able to filter our hits according to the guidelines that they propose. And lastly, we will be in discussion for productization with our pharmaceutical partners. And with that, I would like to thank everybody who is here to listen to my talk, and I'd be very happy to engage you all in the breakout session.